Dear Board of the BGPN, I would like to express my sincere gratitude towards you for nominating my PhD dissertation for the doctoral award within the field of psychiatry. I am truly humbled by obtaining such recognition from experts within the field, so thank you very much for that. I would also like to excuse myself for having to give this presentation in English. I still have to fully master the German language, and unfortunately I am not there yet. So this presentation will be in English. So this is the title of my PhD, an investigation into the repetitive pathophysiology and the effect of a non-invasive targeted treatment strategy in an animal model overexpressing the dopamine transporter. I conducted my thesis in the group of experimental psychiatry at the Charité under the leadership of Professor Christine Winter. My thesis focuses on neuropsychiatric disorders and more specifically on repetitive disorders such as Tourette syndrome. Now in Tourette syndrome, the involuntary movements constitute the core symptoms, and these are the so-called tics. And these tics are often treated with systematically applied therapies such as drugs. However, unfortunately, very often, these drugs lead to insufficient symptom relief and potential side effects. So to move forward, we need to further understand the pathology underlying the repetitive disorders, and we need to develop new targeted treatment strategies that specifically interacts with the processes involved in symptom generation. The pathology of repetitive disorders has long been centered around the corticostriatal circuit, and this is a circuit that under normal circumstances is involved in the regulation of movement. Within this system, Tourette patients display alterations in the dopamine system, and this includes alteration in dopamine levels and dopamine receptor availability. Now, these findings have indirectly pointed towards an overexpression of the dopamine transporter. However, the direct role of dopamine transporter overexpression still needs further investigation. In addition, Tourette patients display a specific decrease in cells within the striatum, and these are the so-called PAV-albumin expressing interneurons. And it is thought that it is the combined action between the alterations in the dopamine system and the abnormalities within the striatum that leads to an imbalance within the corticostriatal circuit, which eventually evolves into a hyperexcitability of the cortex and then to the involuntary movements that we see in the disorder. So to target the pathology, we need to target this circuit. There are a few targeted treatment strategies out there, and one is the so-called deep brain stimulation. This is an invasive procedure where areas considered involved in the pathology are directly stimulated through the implementation of electrodes. Now, in the context of repetitive disorders, there's already been several targets tested, including the thalamus, the globus pallidus, the nucleus accumbens. However, so far, the optimal target still needs to be identified. Another treatment that also has a targeted approach is the transcranial direct current stimulation. This is a non-invasive procedure that modulates the cortical excitability. And here the type of stimulation that you apply eventually determines the effect on the cortex. So if you apply a nodal stimulation, this has a tendency to increase cortical excitability, whereas if you apply cathodal stimulation, this has a tendency to decrease cortical excitability. Now the question here is whether transcranial direct current stimulation, or TDCS, may be used to target the cortical excitability that we see in repetitive disorders and thereby to provide symptom relief. So this leads me to my three primary questions of my thesis. The first one being, what are the consequences of that overexpression? So to answer this question, we needed a model, and here we went for a transgenic rat model that overexpresses the dopamine transporter. This model was constructed in our lab, and as a part of my thesis, I was involved in the characterization of this model, and this was done both on a neurobiological as well as a behavioral level. We named the model the DAT rat, and this will also be the name that I will be using for the remaining of my presentation. Its corresponding control was the so-called wild-type rat. Now, initially, we wanted to see how DAT overexpression affected the dopamine system in these animals. And here we found that DAT overexpression leads to alteration in dopamine levels in specific brain areas. And this was coupled with an increase in monoamine oxidase activity, as well as an increase in dopamine turnover. In addition, DAT overexpression also led to alteration in the expression of dopamine receptors. So collectively, this shows that DAT overexpression, maybe not surprisingly, leads to substantial deficits within the dopamine system. Next step was to investigate the implications going beyond the dopamine system, so how the corticostriatal circuit was inflicted due to the DAT overexpression. Now, as I mentioned in the previous slides, patients display a specific decrease in striatal parvalbumin expressing interneurons, and interestingly, this was also found in our DAT rats, as they showed a specific decrease in these interneurons in the caudate putamen. I also mentioned that patients display cortical hyperexcitability, and this was also found in our DAT rats, as these rats display an increase in CFOS expressing cells, specifically within the orbitofrontal cortex. So collectively, this shows that DAT overexpression leads to abnormalities within the corticostriatal circuit, and this to abnormalities that are also seen in the repetitive pathology. Now we wanted to see whether these neurobiological alterations eventually led to an increased susceptibility towards repetitive behavior. So what we did was that we applied 3 different dosages of amphetamine to these animals and then followed this up by behavioral scoring. And here we found that the DAT rats displayed significant increase in repetitive behavior at a dosage that was ineffective in the wild-type rats. When we then assessed how this behavior developed over time, we could see that the repetitive behavior only started to manifest itself 70 to 90 minutes after injection, so indicating that the development of this behavior was time dependent. We then wanted to test how this behavior responded to drugs that are also used in Tourette patients. So how close the behavior is to what we see in also Tourette's syndrome. What we did was that we applied clonidine prior to the manifestation of a repetitive behavior. And what we found was that this significantly decreased the occurrence of the repetitive behavior in the DAT rats. So to answer the first question, what are the consequences of DAT overexpression? Well, we found that DAT overexpression leads to repetitive pathophysiology, both within the dopamine system as well as the corticostriatal circuit. And these abnormalities translated into an increased susceptibility towards repetitive behavior that was sensitive towards drugs, usually applied in the treatment for Tourette syndrome. Now we wanted to dig a little deeper and figure out which specific brain regions are involved in the repetitive behavior that we see in our DAT rats. So this we investigated by using deep brain stimulation. And here we implanted electrodes in several brain areas, both cortical and subcortical areas. And then we applied stimulation prior to the manifestation of the repetitive behavior. What we found was that repetitive behavior in the DAT rats significantly decreased solely when we stimulated areas of the sensory motor circuit. So this being the primary motor cortex, as well as the caudate putamen. In contrast, repetitive behavior increased when we applied stimulation in areas also found responsible for repetitive pathology. Here we found an increase in repetitive behavior in our control rats. So collectively, this shows that areas of the sensory-motor circuit plays a role in the generation of the repetitive behavior that we see in our DAT rats. The last but not least, we wanted to see what are the effects of a non-invasive stimulation procedure in these animals. So what are the effects of the transcranial direct current stimulation that I talked about previously? So to investigate this, we applied stimulation to the frontal cortex prior to the induction of repetitive behavior, and then we assessed how this then eventually affected the repetitive behavior in the animals. Here we tested different types of stimulation, so both the anodal and cathodal type of stimulation, and this at different dosages. Once behavior had been investigated, we then went a step further and then took a look at the most effective type of stimulation to see how this affected the animals on a neurobiological level. So in regards to the behavior and the different types of stimulation, we found that repetitive behavior was solely reduced when we applied a nodal stimulation to the DAT rats. However, we also saw an opposing effect between the groups because a nodal stimulation actually increased repetitive behavior in the control rats and the wild type rats. Furthermore, we saw that this effect was only obvious when we applied 200 microamps. So collectively, this showed us that the effects are state dependent because we see a differential effect between the two groups. And also it shows that the stimulation is polarity specific since only a nodal had an effect, and that it follows a non-linear dose dependency. Now we applied TDCS to the frontal cortex, yet this constitutes a relatively large cortical area. So to pin down and to better get an idea on which cortical regions were especially altered following the application of a nodal TDCS, a computational model was constructed, and here we found a prominent peak in the current density over the region corresponding to the motor cortex. So this shows us that the motor cortex may be a potential key region when it comes to the therapeutic effect of a nodal TDCS in modulating the repetitive behavior that we see in our DAT rats. And this correlates very well with our DBS findings, showing that the sensory motor areas are involved in the modulation of the repetitive behavior in our DAT rats. Now I showed you on the previous slide that the DAT rats display substantial deficits within the dopamine system. So we also wanted to see whether the effects of a nodal TDCS involved a regulation of the dopamine system within these animals. And here the results showed that DAT rats continued to display a decrease in dopamine levels as well as an increase in dopamine turnover. So this showed us that the effects of a nodal TDCS went beyond the dopamine system, so something else was going on. Zooming out a bit, we wanted to see how the corticostriatal circuit was affected by the application of TDCS. And here we found a modulation of mRNA parvalbumin levels in the striatum, as well as an alteration in mRNA levels of CFOS in the cortex, both in the medial prefrontal cortex and the orbital frontal cortex, showing us that the therapeutic effect of a nodal TDCS may involve modulation of both cortical and subcortical areas of the corticostriatal circuit. So to answer the last question, what are the effects of TDCS in the DAT rats? Well, we found that only a nodal stimulation reduced repetitive behavior in the DAT rats, and that this effect was state dependent and followed a non-linear dose response. And this modulation of behavior was further coupled with an interaction with both striatal and cortical activity properties, and that this effect could be appointed to the sensory motor circuit. So to conclude, we found that DAT overexpression is implicated in repetitive pathology and behavior, and that a nodal TDCS reduces this behavior through a modulation of the circuit considered involved in a repetitive pathology. So this sets the stage for further investigating the use of TDCS as a treatment for repetitive disorders. These are the publications that went into my thesis. This is my sincere thank you to the ones who made it possible. So first and foremost, my supervisors, Professor Andreas Heinz and Professor Christine Winter, and also thanks to both the Berlin and Dresden team. And I would like to thank you for taking the time to watch this presentation, and of course, once again, for nominating my PhD dissertation for the doctoral award. Thank you.